We've heard about current therapy for glaucoma. We are really, really fortunate to have with us Dr. Johnson, who's really going to give us a look into the future, which a lot of you are here to hear about, is to see what's on the horizon for us in caring for glaucoma. Dr. Thomas Johnson is the Shelley and Alan Holt Rising Professor of Ophthalmology at Wilmer Eye Institute, Johns Hopkins University in Baltimore. And thank you so much for coming.
All right. Good morning, everyone. Thank you. I want to thank the Glaucoma Research Foundation for the invitation to come speak to you all today. I'm very excited and very happy to be here. In addition to being a glaucoma specialist, I'm also a neuroscientist, and I have a laboratory at Johns Hopkins whose sole focus is on developing the next generation of new and better treatments for glaucoma. And so that's what I'm going to tell you about. I want to give a little bit of a caveat to begin with. I want everyone to be on the same page and let you know that while some of the things I'm going to tell you about are very exciting, they're not going to be available next year or the year after. We're thinking something on the 5 to 10 year time horizon, but hopefully we'll get there together. So the reason I think we're all here is that glaucoma, unfortunately, is a really tough disease. Glaucomatous vision loss is irreversible. Your doctor's probably told you that on the first day that you may have been diagnosed, that while we can slow down or halt the disease from getting worse, we cannot do anything right now to restore vision that's already been lost. And that's important because the disease is the sneak thief of sight. It's a very insidious disease that happens very slowly. People don't notice it. And so a lot of people are only diagnosed with glaucoma after a tremendous amount of vision loss has already occurred. And other patients continue to suffer worsening of their disease despite our best efforts, because eye drops sometimes fail and lasers sometimes fail, and sometimes even surgeries. Patients need multiple surgeries to get them into a stable place. So there are millions of people around the world that have permanent vision loss, and in some cases total blindness from glaucoma. And at this point in time, we can't do anything about that. So the goal of my laboratory is to take the glaucoma profession and transform us from plumbers into electrical engineers and actually develop ways to regenerate vision for patients that have lost it in glaucoma. Now, in order to understand what I'm going to talk to you about, we got to do a little bit of a brief review of what's going on between the eyeball and the brain. As you know, glaucoma is an optic neuropathy, meaning it's an optic nerve disease. And the optic nerve is your main connector that takes all the information about light entering your eye and transmits it through the skull and into the brain. And it's a very complicated wiring pattern. This optic nerve, you can think of it like a fiber optic cable. If you've ever cut open a fiber optic cable, it's got hundreds, thousands, or millions of little fibers in it. And those fibers actually split up in the brain and go to all different sorts of places to allow you to see. Those fibers, though, are all connected to a nerve cell that lives inside the retina in the back of the eyeball. That type of nerve cell I'll refer to as an RGC. It stands for retinoganglion cell. And this is an example of a retina that uses a special dye so that you can see the locations of only about 10% of the RGCs that are actually in this retina. So they cover the entire surface of the retina. They receive information from the other cells of the retina. You've heard of the rods and cones. That's what actually senses light. But the rods and cones need to send that information to the RGCs. And then the RGCs each have a very long fiber called an axon, which head towards the optic nerve right in the middle. And then if you can imagine, the brain is behind this screen. Each of these axons turn 90 degrees and run out the back of the screen in order to get to the eye. If you look at the retinoganglion cells in higher magnification under a microscope, this is what they look like. They've got a little circle in the middle. That's the cell body, the headquarters of the cell. And then they've got all these little finger-like projections, and these are called dendrites, or sometimes I'll refer to them as neurite. These are little projections that reach out to the rods and cones and the other types of cells in the retina and communicate with them. They form communication stations that allow them to receive information about light coming into the eye. And this is a schematic. So if you take the retina and cut it in half, the rods and cones live in the way back of the retina. There's a type of red neuron called a bipolar cell that lives in the middle, and eventually all the information about light is communicated to the retinal ganglion cells up here at the front of the eye, which then have axons that go out the back of the eye. So vision loss from glaucoma occurs because the optic nerve degenerates. And what that truly means is that the retinal ganglion cells are dying. So it's not just that the optic nerve is lost and all the fibers within the optic nerve go away, but the cell bodies that are connected to those fibers within the retina are also gone. And so in order to restore vision for patients with glaucoma, we need to come up with ways of actually replacing the RGC neurons that have been lost. Now, it's a very exciting time to be in ophthalmology and the regenerative medicine field. There's a lot of very cool stuff happening in regenerative medicine, and particularly in the eye. The first United States patients just this past year received stem cell-based treatments with a different type of cell into the retina in order to treat age-related macular degeneration. So we're getting to the point where this kind of thing is happening. We're learning about the safety of how to transplant cells into the eye. But I'll tell you that replacing the cell type that's lost in macular degeneration is a lot easier than what you need to do in glaucoma, because the macular degeneration cells just sit in the retina and do one thing in a very localized manner, whereas the ganglion cells, as I told you, connect to all kinds of other neurons in the retina and have to project all the way to the brain. So this is sort of our roadmap of what we're trying to accomplish, and it is not an easy task. The National Institutes of Health that funds some of this research has labeled this an audacious goal, to be able to take stem cells from human patients, turn them into retinal ganglion cell neurons, figure out a way to transplant them into the eye, get them to survive, and then migrate into the retina, into that film in the back wall of the eye, then get them to send out all those little finger-like neurites so that they can receive information from the retina about the light coming into the eye, then get them to grow axons or fibers that go to the brain and make the right connections there. So again, this is why this is not going to happen very soon, but I think with the right people working on this and the right resources, it can happen in a matter of several years. I'm going to tell you a little bit about our work. This is a picture of some human retinal ganglion cells that we've created in a dish. We start with stem cells that, as you know by now, can be obtained from patients with something as simple as a skin biopsy or a blood sample or even a urine sample. And we can genetically engineer them in a dish to stop becoming skin cells and instead to become stem cells that can make any cell type in the body. And we've worked out ways to direct them to become RGCs, the very type of nerve cell that is lost in glaucoma. And a lot of other investigators have been working for several years on ways to make RGCs more regenerative. When you're during development, when you're in a fetus in utero, the eye is growing and developing, and those ganglion cells are naturally growing processes that find their way to the correct locations in the brain. But once you're an adult or you have a developed eye, a lot of the signals that drive that process are lost. And in an adult, if we have a model of an optic nerve injury and you damage retinal ganglion cells, they die and they do not regenerate. But several investigators at all places throughout the world have found a number of different pathways, genes and drugs that, if delivered to the retina at the time of an optic nerve injury, can actually get axons to regenerate through the optic nerve. That's what these green fibers are. These are individual retinal ganglion cell axons, and this is what happens if you don't give them those treatments. So we now have methods here's another example that we think we can leverage to not just protect the endogenous or the naturally occurring optic nerve, but to actually also apply those to stem cells so that when we transplant them into the eye, they can regenerate to the brain. So the main point I want to make now is that this is not a new idea. People have been thinking about replacing retinal ganglion cells and regenerating the optic nerve for probably 40 or 50 years. But for a long time, it's been considered science fiction because we didn't have the tools and the knowledge necessary to do it. And I believe we really are at a juncture now where stem cell technology, CRISPR gene therapy, electrophysiology and electrical field stimulation, and some of these technologies have come to the point where this might actually be feasible if we can work together and work out a protocol that actually drives the restoration of the optic nerve efficiently. So I want to show you a video now. This is where we started about 4 years ago. The red that you're looking at in this video are the human transplanted retinal ganglion cells. And the blue, which comes and goes in this video, is the recipient retina. It's the retina of the eye that we transplanted the stem cells into. And this is a three-dimensional reconstruction, so you can see what's happening in this volumetric block of tissue. And you can see that those cells survive for several weeks after transplantation, and they grow neurites and processes, but they grow on the surface of the retina. They're not integrating at all into the retinal tissue. And this is a problem because you don't just want new neurons to be lining the back of the eye. In that position, they will receive no information about what you're looking at, what the light coming into the eye is trying to tell your brain. So we needed to find a way to get these cells to actually migrate and integrate into the tissue so that they would look like they're really part of the tissue and hopefully develop the connections with other types of cells in the retina that would allow a signal to be propagated from the rods and the cones to the ganglion cells and to the brain. So part of our work identified that there is a naturally occurring membrane that sits on the surface of the retina. This is called the internal limiting membrane, or ILM. And in early experiments, we found that if we had cut a little nick in the internal limiting membrane, then locally it actually allowed the RGCs to integrate. So this is a top-down microscopy view of some cells. Again, the red cells are the transplanted neurons, and that green sheet on the left is the ILM. There's a break in the ILM down here, and if we computationally just look underneath the ILM, we can find that those RGCs that we transplanted now are able to migrate laterally into the tissue and begin to integrate in the manner that I was saying we want to have done. So first we needed to develop a better way to actually disrupt this ILM membrane on purpose and in a controlled manner. And we developed a couple of ways to do this. One is with using genetically engineered models of transplantation. Another one is just by injecting an enzyme into the eye that's capable of digesting that membrane. And you can see here that when we inject that enzyme into the eye, it makes this membrane look like Swiss cheese. It pokes holes in it that we thought would maybe allow transplanted neurons to integrate into the retina. And when we do that, we were pleasantly surprised to see a few things. So this is another example of a retina where we've transplanted human RGCs. We see that they survive for a fair number of weeks after transplantation, and they actually integrate into the retina. Here, this is a tracing of these finger-like projections, these dendrites put out by this neuron, and it's deep within the tissue, and it's specifically putting out these dendrites within the layer of the retina where the natural communication stations or synapses with the other retinal neurons occur. The other thing you'll notice is that each of these cells is sending out a very long fiber, an axon, that somehow knows where it's supposed to go. Most of these are being naturally directed towards the optic nerve head. We didn't actually think this would be the case. We thought that after development in an adult eye, these neurons would have no idea where to send their axons, but they do. So we were, in some ways, lucky that some of the challenges that we thought we would need to be overcome may not be as challenging as we thought they would. Now, this is another movie similar to the one I just showed you, but this time we've disrupted the internal limiting membrane. And you're going to notice a difference here about how these cells interact with the underlying tissue. So this is one cell right there. We've traced its finger-like dendrites so that you can see very clearly where they are in the tissue. And now if we spin this around, you can see that this neuron is within the tissue itself. It's migrated in, and it's in the correct layer. This gap, this layer up here in between the two lines of blue cells is called the inner plexiform layer, and that's exactly where the connections between bipolar cells and RGCs occur. So now we are not only transplanting RGCs into the eye that survive, but they send their listening ears, their dendritic projections, into the correct part of the retina in order to receive information about the images that are entering the eye. Now, we've been doing this in several different models, but obviously the end goal of all of this is to transplant RGCs in human patients with glaucoma. So it was important that we validate all the findings that we have in tissue from human people. Now, obviously, we're not at a stage yet where it would be safe for us to do this in living patients as a clinical trial, but we have obtained eye tissue from human tissue donors. If these eyes were not healthy enough to be used for things like corneal transplant, then they're donated to science. And when we've transplanted human retinal ganglion cells onto the retinal tissue and cultured them in an incubator for a week, we noticed the same trends, that if you do not disrupt that membrane, there's very little integration with the retina. But if you disrupt the membrane of the eye, then you get much better engraftment of the donor neurons into the eye. So what are we doing now? Well, we're trying to go even further to preclinical models that are much more similar to human patient. So these are eyeballs that are the same size as human patients, and we can do things in these that are not able to be done in, for instance, mice. In this case, you're looking at a surgery of a human patient where Dr. Jim Handa, a vitreoretinal surgeon at Wilmer, is peeling the internal limiting membrane off of the retina. Now, this is actually done routinely for treatment of a whole different separate set of diseases like macular holes and epiretinal membranes. But this shows that it's not only safe and feasible, but routine in some ways to surgically remove the ILM. And so we've done that in this model with a surgical strategy that could be directly applied to human patients. This was done in an ophthalmic operating room with all the normal tools and strategies and resources that a human surgery would involve. And when we transplanted these cells into eyes where the internal limiting membrane was peeled, we got very good integration of these cells into the retina. And just like in the other examples I was showing you, they grew axons going directly towards the optic nerve head. So this provides very good rationale that we're able to get the cells at least connected in the retina at this time. And this is, I'm going to skip over that. If we could play this video, I think this is the one that I can't play on my own. The most recent thing that we've done, and what I think is the most exciting, is a little bit of a complicated experiment, but I'll walk you through this. To remind you, we've shown that when we transplant RGCs, they send their dendrites into the correct location of the retina, where in theory, they could begin receiving information that ultimately comes from the rods and cones as they sense light. But we wanted to prove that they're actually receiving information, that they're not just in the right place, but that they're functioning properly. So in order to do this, we had to genetically engineer our transplanted stem cell-derived neurons with a special gene. And that gene is a fluorescent protein that glows under the microscope, but it isn't stable in its brightness. It actually will become brighter or dimmer in real time according to how much that cell is receiving information from other neurons. As neurons begin talking to each other, they have electricity inside them that goes up and down, and this gene will glow in a brightness that correlates with how much electricity is going on in that cell. That tells us how active it is. you can see this movie. So we've taken a movie, a microscopic movie over time, and at 10 seconds, a whole bunch of those retinal ganglion cells become very bright, and then they get dark again. And that's because right before the 10-second mark, we flashed an image at the retina to show it to the photoreceptors. And if these cells were not actually receiving that information, they'd be black the whole time. But because they flash, that tells us that the photoreceptors, the rods and cones, are actually seeing that image, and then they're transmitting it to the ganglion cells, and they're becoming electrophysiologically excited because they've received that message. So where we are now is, I think, a tremendous advance from where we were just 5 years ago. We are well on our way to replacing the connections that occur inside the eyeball. And what remains is figuring out how to get those fibers to grow through the optic nerve and into the correct locations in the retina. I want to end with just a word that this is not something that could be done by just one laboratory. Just at Wilmer alone, we have a team of several faculty members that are all contributing to this. And we founded 2 years ago something called the Retinoganglion Cell Repopulation, Stem Cell Transplantation, and Optic Nerve Regeneration Consortium, or RESTORE. There's a QR code if you want to look at the website. But we now have 220 scientists and clinicians worldwide that are collaborating on this problem. We've had 2 conferences so far, and we continue to work together to bring this to fruition. So with that, I want to thank you all for your attention. When we have time for questions, I'll be happy to answer them. And thank you all for listening.